Assalamualaikum and salam sejahtera. Welcome to topic 5 with the title of Breeding Techniques for Ornamental Fish. Before I proceed, let us look at the learning outcomes of this topic. At the end of this topic, you should be able to first explain the general principles of fish breeding. Second, describe reproduction strategies. And the third one, choosing and conditioning the parents. One of the most important aspects of any living organism is to produce new individual from the existing one, thus ensuring the continued existence of the organism concerned. Like other higher animals, fishes are also differentiated into males and females. The male producing sperm to fertilize the eggs of female, which develop into new generation. Most of the fishes release their eggs and sperm in water, and fertilization takes place externally. Some fish species, however, have developed a system of internal fertilization where embryonic development takes place inside the body cavity of the fish. The culture of ornamental fishes is called as aquariculture. For your information, breeding aquarium fish is one of the steps to becoming a skilled aquarist. Once fish are kept healthy enough to spawn, the novice knows that they can attempt to keep more challenging fish. In order to breed a species, the aquarist usually needs to be able to distinguish between the sexes and to be able to recreate natural conditions to stimulate spawning. Always record your successes and failings in a notebook. What are the general principles of fish breeding? Most fish kept by aquarists are oviparous, but there are some exceptions, such as viviparous fish. A varied and rich diet enables fish to accumulate reserve of proteins and lipids, which help them form eggs and spermatozoa. This is the maturing phase. When the female's ovaries swell, she is ready to lay eggs. This only needs a stimulus to trigger it off, such as in the natural setting, rain, sunlight, or moonlight. There are several factors affecting reproduction. First, the environmental factors such as light, temperature, water quality parameters, and etc. The second factor is intrinsic physiological factors such as hormonal and neurohormonal. The third factor is nutritional requirements of fish. These include natural food, feeding pattern, and balanced diet required for optimal gonadal growth. In terms of sexing fish, most fish can be classified as sexually dimorphic or sexually isomorphic. In sexually dimorphic species, the sexes can be easily distinguished by the primary characteristics such as the shape of the sex organ, the secondary differences such as the size, shape, color, and finish. Generally, males are frequently more colorful, larger, and have more elaborate finish. In sexually isomorphic species, there are minute, if any, apparent sexual differences. Often, the only way to distinguish between the sexes is the shape of the genital papilla, which is only visible around spawning times. Some sexually isomorphic species have no known external sexual differences. There are several criteria that you need to consider when selecting the parent fish. First, choose fish that display good markings and color. Second, only use mature, healthy fish for spawning. Third, be sure that the pair is compatible. Avoid crossing different strands or color forms because the young are often unattractive. Last but not least, make sure that the pair are both of the same species because hybrids are sterile. With some cichlids and killifish, females of different species look similar. Now, let's move on to the reproductive strategies. The majority of aquarium fish are egg layers with external fertilization. Egg layers can be divided into 5 groups, which are egg scatterers, egg depositors, egg barriers, mouth brooders, and nest builders. We are going to look at on egg scatterers first. The egg scatterers include danios, barbs, rasbora, tetras, goldfish, and carps. These species simply scatter their adhesive or non-adhesive eggs to fall to the substrate, into plants, or float to the surface. These species do not look after their brood and even eat their own eggs. These often schooling fish may spawn in groups or in pairs. Often, there is a large number of the small eggs laid. The fry hatch quickly. Now, we are going to learn fish that is under the category of egg scatterers having adhesive eggs. Goldfish is an example of egg scatterers having adhesive eggs. Goldfish is oviparous. It lays eggs. The female releases eggs and the male ejects seminal fluid, which fertilizes. Then a period of incubation follows, after which the baby fish hatch out. Goldfish mature when 6 cm long. 2-year-old males and 3-year-old females serve ideally as spawners. The sexes can be identified on the basis of following characteristics. First, belly line. With the male, the belly line continues from the lowest point of the belly to the base of the tail in a slight curve. But with the female, the line is almost circular when viewed from the top. Second, genital orifice. The shape of genital orifice in male is smaller than that of the female and is of a long oval shape, while that of the female is round and slightly protruding. Third, tubercles. As a secondary sexual characteristics, during the mating period, the males develop small white tubercles on body tissues, also referred to as pearl organ or pimples, on its operculum and pectoral fin, and sometimes on other fins as well. The number of dots is set indicates the level of male spatial. For developing the broodstock, healthy fishes in the age range of 8 to 15 months, weighing 40 to 100 grams, are carefully selected out of the parent stock in the ratio of 2 male to 1 female and maintained sex-wise in semen tanks. Feeding is done both in the morning and evening with live feed and artificial feed. When the coursing stars appear on the body of males, it is indicative of the fact that the time has come for spawning. Before taking up the breeding, tanks are disinfected with 1 ppm solution of potassium permanganate, washed thoroughly, and then filled with fresh water. Since goldfish eggs adhere to surfaces, prepare some sort of nest for them. Various types of water plants can be used for this purpose. Since it is possible that some other kind of fish or aquatic organism has spawned earlier, the plants should be placed in the spawning area at least 10 days before spawning, so that all other eggs will be hatched and eliminated from the plants for the nest. Willow roots and the bark of hemp palm, plants with very small slender branches such as hydrilla or with fibrous roots, can be used. The nest should float close to the surface of the water, but an additional nest should also be spread on the bottom of the spawning area for those eggs that sink instead of adhering. Breeding tanks should be provided with continuous flow of running water through sprinklers to induce releasing of eggs and also to maintain the temperature between 20 to 31 degrees Celsius. After conditioning, the male and female in the ratio of 2 to 1 are released into breeding tank in the late evening hours. Egg laying usually takes place within 6 to 12 hours. The male begins chasing the female in a crazed manner. The male presses its operculum against female's abdomen and fertilizes her eggs while swimming beside her. As soon as spawning is completed, the nest is transferred to a different container or the parent fish transferred away from the nest. If this is not done, the parent fish will eat the eggs. Each female lays about 2,500 to 3,000 eggs. Healthy eggs are transparent at first, but as development progresses, the transparent area decreases. Unfertilized eggs, or those in which growth has been stopped, will always remain transparent. Dead eggs become cloudy white and hair-like aquatic fungus will grow out of them. The period of incubation differs according to the water temperature. The warmer the water, the less the number of days necessary for hatching. When temperature is too high or too low, deformation in tail shape is common. Under ideal condition, within 3 days, the eggs hatch out with a hatching rate of 80 to 90%. When the larvae begin to float, the nest materials are taken out from the tank. Sometimes, infant fish remain still clinging to the nest, so care must be taken to get them out of the nest before removing them from the water. Rearing of larvae is carried out in larval rearing tank. Soon after the larvae have absorbed their yolk and begin swimming around, it is necessary to give them food. First feeding of larvae is done with infusoria. This continues for 7 days. In the second week, infusoria is supplemented with small quantity of boiled egg yolk and microworms. From 3rd week onwards, Daphnia and tubifex are provided till the babies attain 10 to 15 mm length. After about a month of rearing, the young ones, or known as fries, are transferred to grow out ponds. After 90 to 120 days, when young ones attain the marketable size of 5 to 10 cm size, they are transferred to holding tanks where from they are marketed. Next is egg depositors. The egg depositors include Raspora, angelfish, cichlid, and some catfish. These species deposit their eggs on a substrate such as tank glass, wood, rocks, and plants. Egg depositors usually lay less eggs than egg scatterers, and the eggs are larger in size. Egg depositors fall into 2 groups. First, those that care for eggs. These fish form pairs and the eggs are defended and clean. Second, those that do not care for the eggs. Egg depositors that care for their young can be divided into 2 groups. First, cavity spawners that lay their eggs in a cave. Second, open spawners that lay their eggs on an open surface. The eggs take a few days to hatch, and the fry are often gathered by the parents. Depending on the type of egg depositor, the tank should be furnished differently. For those egg depositors that care for their young, the parents can remain in the tank after spawning. For substrate spawners, depending on the species, it should be given tanks with furnished glass pans, broadleaf plants, or flat stone as spawning sites. Some species, such as discus and angelfish, prefer vertical surfaces. For cavity spawners, flower pots turned on their side, coconut shell, and rocky cave are suitable spawning sites. The tank should be furnished with either live or plastic plants to give the fish a sense of security. One of the examples of egg depositors is Raspora. To breed Rasbora, a slightly warmer temperature, around 80 degrees Fahrenheit, will help to encourage spawning. Rasbora fish will need nutritional food full of protein too, such as daphnia and bloodworms. Keep 2 females for every male. These males will perform a display to entice the female to mate. You will need some broadleaf plants in your tank because once ready to spawn, the female rubs her belly on the underside of a leaf to signal the male. The male will come to fertilize the eggs. This will be attached to the leaf and hatch after 24 hours or so. Separate the parents from the fry as they will try to eat them. The fry will be nearly completely clear and feed on a yolk sac for the next 24 hours. After this, you will have to start feeding them. Newly hatched brine shrimp are one of the few food small enough to give them. Next reproductive strategy is egg barriers. The majority of egg barriers are annual killifish, which lay their eggs in mud. These species usually inhabit waters that dry up at some time of the year. The parents mature very quickly and lay their eggs before dying when the water dries up. The eggs remain in a dormant stage until rain stimulates hatching. Killifish is an example of egg barrier. Killifish laying their eggs in the soft pit at the bottom of the tank. In an aquarium, they have the habit of laying their eggs in dense plant growth. They are jumpers and therefore kept in covered aquarium. Their eggs are capable of staying viable after being dried and hatching after an interval of some weeks or even months when placed again in water, a provision to tide over periods of drought when the pools dry up and the parents die, but the eggs remain fertile until the next rains. In fact, in some cases, the eggs will not hatch unless they are kept dry for some period. Being insectivorous in nature, they are used in malaria control by being introduced in stagnant water pools. They rarely grow over 3 to 4 cm in total length and are short-lived. Next reproductive strategy is mouth brooders. Mouth brooders are species that carry their eggs or larvae in their mouth. Ovophile or egg-loving lay their eggs in a pit which are sucked up into the mouth of the female. The small number of large eggs hatch in the mother's mouth and the fry remain there for a period of time. Larvophile or larva-loving lay their eggs on a substrate and guard them until the eggs hatch. After hatching, the female picks up the fry and keeps them in her mouth. Paternal mouth brooders. The male looks after the eggs, include the arowana fish and the mouth brooding betta, whereas biparental mouth brooding occurs where both parents take some of the eggs, such as cichlid genus Xenotilapia, the spatula bubble catfish. Next, we have nest builders. Nest builders build some sort of nest for their eggs. The nest is usually in the form of bubble nest formed with plant debris and saliva-coated bubbles, such as library fish and catfish, or an excavated pit in the substrate, such as by cichlids. Nest builders practice brood care. For example, Garamis is a nest builder. Blue gourami breed during April to August. During breeding season, mature males develop dark coloration and females show bulging abdomen. While making breeding pair, care must be taken to select a mature female which is ready to spawn. This is because males of blue gourami are very aggressive in nature and tend to kill a female if she is not ready to breed. 1 or 2 pieces of floating plants may be floated on the water surface. This helps in holding the bubble nest. The tank should not be provided with any bubble aerator. The ideal water condition for breeding is water pH 7 to 7.5, temperatures 26 to 30 degrees Celsius, and hardness 100 to 220 ppm. The pairing of blue gourami is made in the ratio of 1 to 1. The nest filled with saliva of the fish floats at the surface or under plant leaf and looks just like soap form. After the bubble nest is ready, breeding takes place and eggs are lodged in the nest. After breeding, the female is removed as the male takes the honors of guarding the eggs, which remain attached to the floating bubble nest. Hatching takes place within 24 hours. As soon as the fry begin leaving the nest, the male is also removed from the tank. After 36 hours, when the fry are at free swimming state, they are provided infusoria as feed. After 6 to 7 days, the fry start taking brine shrimp or small moina. At this stage, they should be fed 3 to 4 times a day. The growth of fish is very uneven and often some shoot fry develop. The shoot fry, which refer to the bigger ones, should be separated and reared in different containers. Now, they can be diet of worms and stocked in bigger tanks and given a formulated feed. Next, we have life bearers. Life bearers are fish that bear live young. There are two types of life bearers: ovoviviparous, where the eggs form and hatch within the female before birth. Second, viviparous, where no eggs are formed and the young are nourished through an umbilical-like cord or from secretion by the female. Life bearers are often prolific, easily bred species. Guppies, platies, mollies, and swordtails are the commonest examples of life bearers. Adulthood in life bearers is reached as early as 6 to 8 weeks after birth in guppies and platies, while mollies take double this time to mature. In order to fertilize the female, the anal fin of male gets developed into a structure called gonopodium. This is used to insert the packet of sperms into the female fish. Once fertilized, a female can produce several broods from one mating, since she is able to store male sperm in her body for several months. The male spread its fins and quivering, keeping his body parallel to the female, and a little behind her make a quick thrust with his gonopodium and swim away. In this egg, sperms are introduced in the female's body and the eggs are fertilized. Development of baby fish takes place inside female's body, and after about 4 weeks, a brood of 6 to 100, depending on the species and age of the mother, is delivered. In guppies, the number of babies in a brood may be 6 to 60, mollies up to 100, while a large swordtail may deliver as many as 200 babies. It is easy to know when a female is about to give birth by spotting the dark mark called gravid spot located close to the urinogenital aperture. A female is pregnant can also be known by the bulging appearance of her abdomen. As the female life bearers have the habit of eating their own newborn young ones, therefore for breeding they are required to be kept in special breeding tank. The selected female showing bulging abdomen and gravid spot near the vent are kept alone in a spacious tank with a dense stick of plants at one end. The young ones in mother's belly remain in a folded position. Now sooner they are ejected out. Babies straighten out and swim away to the shelter of the plants. As the newly laid babies are large enough at birth, they can be fed on microworms and brine shrimp nopli. Now, let's move on to the conditioning of the parent fish for breeding process. Before placing the parent fish together for spawning, they should be conditioned. Conditioning is a process of feeding the fish a variety of healthy foods to get them in top condition for spawning. Many species can be conditioned using a well-balanced flat food, though others should be conditioned on live foods such as brine shrimp, insect larvae, and flying insects. The parent fish can be separated while conditioning. This way, when the fish are reintroduced, they are eager to spawn. Before we end our lecture, let us recap on the subtopics that we had learned. We learned on the general principle of fish breeding, several reproductive strategies, criteria in choosing the parents, and ways of conditioning the parents. With that, thank you for your attention. Until we meet again in our next topic.